Genetics, Faculty of Agriculture, Asyut University, Egypt, under the patronage of Professor Ahmed El-Minshawi, President of Asyut University, Professor Jamal Badr, Vice President for Graduate Studies and Research, Professor Adil Wannan, Dean of Faculty of Agriculture, Asyut University, Professor Mohammed Saghir, Vice Dean for Graduate Studies and Research, and Professor Ahmed Salam, Professor of Genetics Department, Asyut University. I am Said Mohammed. I am Associate Professor of Genetics at Genetics Department, Asyut University. I am the moderator for this seminar today. Our speaker for today, Dr. Mohammed Ali. Dr. Mohammed is Associate Professor of Group Genetic and Breeding at Department of Genetic Resources, Desert Research Center in Egypt. Dr. Mohammed received his PhD in 2018 from Haozang Agricultural University in China. From 2008 to 2024, he has working as a researcher in Desert Research Center. He has been working in plant biotechnology in the field of plant functional genomic and secondary metabolism for 16 years. His current research involves in the usage of different plant metabolites database and the new techniques such as RNA sequencing. Dr. Mohammed published many peer-reviewed papers in top scientific journals. He also works as a certified reviewer and guest editor for many important scientific journals. Dr. Mohammed's presentation today entitled Induced Genetic Diversity through Mutagenesis in Wheat Gene Pool and Significant Use of SCoT Markers to Underpin Key Agronomic Traits. He has 13 minutes for his presentation and another 15 minutes to answer our question. Dr. Mohammed, you can start your presentation.
Thank you, dear Professor Syed, and great thanks for my dear Professor Ahmed Salam and Dr. Abdullah and his team for this kind of invitation. Our title of our presentation today about induced genetic diversity through mutagenesis in wheat gene pool and significant use of SCoT marker to underpin key agronomical traits. As we know, wheat is one of the most important food crops over the world. In Egypt, wheat is one from the oldest and the most kernel crops that used for Egyptian people. These slides show about distribution and the top wheat producer country in the world. This is about 2020. The biggest country for producing wheat in the world, starting from China, followed by India, Russia, United States, Canada, France, Ukraine. And this slide's visualization about the wheat export by country. We found about Russia is the biggest country for producing wheat in the world, followed by Canada, and then USA, followed by Australia, France, Ukraine, and Argentina. Uses of wheat in food. Wheat is a major ingredient in such food as bread, cakes, biscuit, pasta, noodles, pizza, and other else. We talk today about our article, and this article have the main of our article today is to study conclude as the impact of mutagenesis on the genetic diversity of the wheat gene pool to enhance the agronomical traits through enhancing SCoT marker to identify and characterize it, improve phenotypics expression of some important agronomical traits in wheat. Secondly, to study the correlation between the SCoT marker and the specific agronomical traits, provide insight into the genetic mechanism underlying mutagenesis induced change in wheat. Thirdly, we using in this study some database to predict the mutative tissue expression of our molecular markers, SCoT primer, okay, and determine this is SCoT primer related with which genes, okay. Material and the method of our work, okay. As we know, there is several tools for genetic improvement of crops, starting from mutation, followed by the hybridization and the gene transfer and gene cloning. This is a different method. Today, we talk about the mutation. Okay, mutation, definition of mutation in biology. A mutation is alteration on nucleic acid sequencing of genomics of an organism. And some scientists use mutation as a cheap and cheapest method for increasing genetic viability and enrich plant germplasm for direct selection and crossbreeding. Mutation breeding is complementary method to traditional wheat breeding techniques and utilized method for induced mutation usually in the seeds. There is two ways for induced mutation using radiation, and the other method used mutagenesis, chemical mutagenesis, such as sodium azide, nitrose ethylurate, ethyl methane sulfonate, cholecystine, hydrazine hydrates, nitrose methylurate, dimethyl sulfate, and nitrose alkyl urate. After we use chemical for induced mutations, there are several ways for evaluating the mutation lines. This is based on morphological and agronomical traits, biochemical traits, physiological, DNA-based markers, cytogenetics. In our study, we used the three Egyptian genotypes, CITS1, CITS12, Giza 168, and these three genotypes originated from Egypt, and this table shows the degree of each genotype that we used in our study. This investigation was carried out in Botanical Department, Faculty of Agriculture, Al-Azhar University, a SCoT branch during the period from 2080 to 2020. This is a start method that already we get approximately 100 grain with good healthy and good viability for every treatment, and we're starting to immerse in distilled water for about 10 hours. After that, we get the seeds and we re-soak it in the different concentration from sodium hydrazate and hydrazine hydrate for about 12 hours. Okay. After that, we collect the seed and try to wash on the or rinse this seed by distilled water for several times, about 2 hours. Okay, and then we start to growing the treatment and anti-treatment seeds which used as a control in this study. Okay, to planting in the soil. The experimental plot was about 2.5 meter long and 20 centimeter apart, and was assigned under random complete block designs. Okay, for the first generation and second generation. Okay, and this table show about the different concentration from sodium azide and hydrazine hydrazide. This is starting from 0.01 to 0.03, and in each generation we measurement the morphological and agronomical traits based on plant height per centimeter, number of spike per plant, spike length in centimeter, number of spikelet by spike, number of grain by spikelet, and the weight of 100 grain. As you see here, this is a variation in the between the original or the mother genotypes and the other mutant lines that we already select. For example, in CITS1, we select about 3 mutant genotypes: CITS1 36, CITS1 83, CITS1 107. And from CITS12, we select about 4 mutagenic lines. For example, 129, 145, 167, and 183. And from genotype Giza 168, we just select 1 mutant genotype, 280. And as you see here, there is a variation about the size and the number of the seed, and also the shape of the spike and the liquid, the grain and the spike. And in this table, we show about the main performance of 11 genotypes that we already, that's including 3 mutant genotypes and other mutants that we already selected from each one. After that, we start evaluated of genetic diversity by SCoT markers. A SCoT marker, as we know, this is abbreviation of start codon targeted polymorphism marker. A targeted fingerprint marker techniques has gained considerable importance in plant genetic and genomics and molecular breeding. And we used these markers. This is in large area and for distinguishing about genetic variation in specific genes that link it with specific traits. And in this case, we used 9 SCoT marker, and we start to design primer. And if this is some of this primer is already published before in other article, we collect also from NCBI. We have, and this is table number 2, show about the primer code and the sequencing of each primer and the annealing temperature. And below the table of number 2, this is a condition for PCR we already used. And the table number 3 show about data about polymorphic and monomorphic band. And as you see here, T represent about total band, and the M represent about monomorphic, and the B polymorphic band, number of monomorphic and the number of polymorphic band. And the percentage B show or present the percentage of polymorphic bands that already get. And as you see here, this is gel electrophoresis represent about the bands for each primer that already used. This starting from SCoT 11 primer number SCoT 11 until SCoT 20. After that, we do about the dendrogram of 3 cultivars and 8 selection mutant line to study the genetic similarity degree. And as we see here, this is dendrogram to show about the group of or similarity between the genotypes, the master genotypes, and between the mutant lines that we already select. Okay. From this conclusion, we found the genetic similarity degree was assigned to be between 0.94 to 0.57, indicate a significant degree of approximately. The adjusted degree of genetic redundance was observed within CIS12 and their mutant line, such as CIS12 145, CIS12 167, and CIS12 129, with similarity value about 0.94. After that, we use the bioinformatic tools to assignment the function of each SCoT, and this is SCoT primer associated with which gene, and this is big problem for some breeding. Just use some techniques, some molecular marker techniques like SSR or CNB or ISSR or other or SCoT, but they in some cases get variation, but they didn't know this is variation between this band and between the mutant and the mother genotype. This is coming from where? So we focus on our study to found the link or associated gene with this primer. Okay, for that we use different database such as Ensemble, Phytosome, National Center of Biotechnology Information, NCBI, Interpro, and the KIG, Qt Encyclopedia Gene and Genomics. Okay, and we are lucky because we found the 9 primer, SCoT primer belong to the same gene, and this is gene related about abscisic acid intensive 3, this transcription factor, and this transcription factor, this kind of transcription factor are characterized by present of 2 main domains, B3 domains and the AB2 domains, and there are potential link to vital biological functions such as responsibility to environmental stresses and to controlling plant growth and development. Okay, and this is transcription factor already characterized in several plants like Arabidopsis, Sariana, Zea, Maize, Oryza sativa, Glycine Max, Solanium, Lycobrisca, Medica justiva, and Capicium annuus. This is a logo for all the database that already used, as we mentioned before, about NCBI, Interpro, Uniport, KGG, Ensemble, and Phytosome. This is the example how we select this gene or know about the associated primer. This is related with which gene. For example, we started to collect several genomics, okay, from all the data as we mentioned before, and download and searching about analyzing data, okay, and used in the sequence for each primer to search about one by one. Okay, we use the forward and reverse primer to search about everyone, and they found this is a high similarity, similarity between the primer, the sequence of our primer and the gene, and for that we select the gene and we get the ID for each gene. Okay, sometimes when you search about NCBI, you found draft genome okay, draft sequencing. We use also the same method, but in this time we divided the whole sequencing to small branching, each branch or several pieces, and each pieces consist minimum about 3 KB, okay, and they use each one for search about other database like phytosome and ensemble to found this is sequencing belong to which gene in related to which gene in wheat genomics. Okay, so after that we select about sequencing and know this sequencing belong to which gene, and for that we get the ID for each gene as we will see here. Okay, for example, SCoT number 11, this is this is ID of genes that the sequence of this primer belong to or have high similarity or has high similarity with this is genes. Okay, so after that we select all the ID for each gene. Okay, after that we use the other database like BAR database. This is the bioanalytic resource data visualization. Okay, and we used about tools related about wheat AFV browser. This is the electrophoresis pictograph. This is data show large amount of data in pictograph, okay, and give us the value of putative expression for each tissue that we already get, okay. And maybe you will be easy to use if you follow my account ResearchGate. You found one presentation talking about every step and how to use this database. Okay, so for that we click about the go button, and this is tools appear like this, and in the primary gene ID we paste the ID that we already get about, for example, about the SCoT primer number 11, and then we start to go and we use the from data source about development atlas. Okay, so this is give you all the stages and all the tissue and all the organs of the wheat. Okay, so from that we, as you see here, after search we get this. This is starting from yellow color to dark red color. Dark red color meaning about this is genes highly expressed in this tissue, and the yellow color represents this is low expression. As you see here, the first gene is highly expressed. This is all of this is expressed putatively. Okay, this is highly in the spike and the grain and the root and the nodules and the shoot apex and the shoot meristem. Okay, as you see here, I don't know this can be here. As you see here, this is highly expression spike. Okay, and this is ovary. Okay, and this is one grain, and as you see here about shoot meristem and the shoot apex. Okay, and this about cotyledon. Okay, we take another example about the other genes. Okay, as you see here, this is the gene, for example, highly expressed in the green endosperm and the embryo. Okay, and the green, and that is only. Okay, so this is explain why this mutation is very important and why this is the effect, and also explains the effect of this is sodium hydrazate, this chemical agent such as sodium hydrazate, sodium azide, and hydrazine hydrate. About effect about why in the figure, or as the figures that already showing before give you a large number of seeds and the seed size and also spike. Okay, after that if you go to the bottom of the window of this windows, we'll find every tissue and have the putative value about represent about the expression about this. Okay, so we collect all these values and put this together in Excel sheet and arrangement all of this data at this form. After that, we use some programs or some software to drawing the heat map and to collect that, as you mentioned here, as you see here. Okay. So this is tell us how to present this data. And after that, we go to mining to understand the effect or the chemical or the mechanism of this is mechanism of action of this is sodium hydrazate, sodium azide or hydrazine hydrazate, and the effect. Okay, we found this is a kind of chemical can can induce mutation through alteration the nucleotide or disrupting DNA replication or introducing during DNA replication and cross-linking to DNA molecules, resulting in single nucleotide polymorphism (SNP). Okay, this is effect also another way explain about other theory, explain why this is chemical that already used in agrochemical as a herbicide. This is, can be also affect about certain checkpoints such as nucleotide expression, repair and repair excision repairing. Okay, and necessary for this is cell to repairing mutations. This is kind of two enzyme that used to repairing the damage and the problems that happen in the nucleotide sequence. So another story to explain why this chemical can induce how this chemical can induce these mutations. This chemical can be disturbed or have a make problem with this as two enzyme. So this is can be creating some kind of signal and nucleotide polymorphism, and this is at final this can be effect about the protein expression. So this can give us this kind of mutation. Thank you.
Thank you for following us. We sincerely thank you for your continued support and engagement across all our platforms. Your trust means everything. Stay connected. Together, we grow.